everyone, today I'm going to talk about DNA and RNA. How do we find DNA? Well, the first people named Frank Griff, okay? And Griff, when he's studying about R-strain bacteria and S-strain bacteria. And now as you can see this picture, the S-strain bacteria can kill mice. And the R-strain bacteria do not kill mice. But if you kill the S-strain bacteria, and the mice won't die. But if you mix the dead S-strain bacteria with the R-strain bacteria, the mouse die. So the group think there's something that will transfer from S-strain bacteria to R-strain bacteria. And another people named Oswald Avery. In 1914, he just inactivated a lot of substance in the S-strain and R-strain bacteria, and he found only the DNA mixed with the R-strain bacteria can develop S-strain and R-strain bacteria. So he said that DNA is a substance which controls the genes. Another people is Hersey and Chase. We know that Hersey is a thief and Chase is a police officer. So Chase chased the Hersey. Okay, and in 1915s, there's two scientists named Hersey and Chase. Okay, and they put the virus in bacteria like that, and they just labeled DNA and a protein in the virus, right? And they found the DNA like that, this picture. DNA go into the bacteria and become a lot of virus. So he knows that DNA is the things that control the genes too. Okay? And what is DNA? DNA stands for deoxyribonucleic acid, which is made of a long chain of nucleotide. And nucleotide is made of three main parts. The 5-carbon sugar, the phosphate group, and the nitrogen containing base. Okay? DNA structure. We know DNA have two strands, double helix, and the strand is made of 5-carbon sugar and phosphate group, while the nitrogen containing base is a genes. It's a genes. What's nitrogen containing base? This is genes we know, and there are 4 kinds of nitrogen containing base: guanine, thymine, cytosine, and adenine. Okay, and A means adenine, and G means thymine, and T means but A means adenine, and G means the A means wait a second. A means adenine and the G means guanine, T means thymine, and C means the cytosine. Okay, and we know that A always pair with T while G always pair with C. And A and T have 2 hydrogen bonds while C and G have 3 hydrogen bonds. DNA replication. Okay, DNA replication is in the synthesis phase, and we know that the enzyme just separates the double helix into a single strand, right? And then the original strand is called the parent strand. It's like a template to make the daughter strand. If the original strand is ATG, the template strand will be TAC. It's a new DNA strand. And one old DNA strand will pair with a new strand. Okay, so it's called semi-conservative replication. So what the central dogma of a molecular biology is from DNA to RNA, RNA to protein, and make protein. Okay, DNA has the instructions for the protein how to make, and however, DNA cannot come out the nucleus too fast. Okay, so but the protein is made at the ribosome in the cytoplasm, so DNA is in the nucleus, we know. So they need the RNA to bring the instruction to the ribosome and make the protein. What is RNA? RNA is like DNA, which is made of nucleic acid. in order to be smaller, so it's not really fat. Okay, and RNA have only one strand, not like double helix, and RNA has uracil instead of 5. Okay, 3 types of RNA: messenger RNA, which is mRNA, which carry the instruction from the nucleus to the cytoplasm. Ribosome RNA or rRNA helps to form ribosome. Transfer RNA or tRNA brings amino acids to ribosome and make protein. So this is looking. What is protein synthesis? Okay, and amino acid with and in protein synthesis, there's only 2 things: transcription and translation. What's transcription? Transcription is the first part of the central dogma of the molecular biology. Okay, and it takes place in the nucleus. And it uses DNA as a template to make RNA, like this picture. So, what is transcription again? And inside the transcription, we know that because RNA can take too much instruction, so they remove some things called the introns, with something that don't have genetic code, and they but they need the exon with the things that have the genetic codes. And then it's just like last picture. And the translation. What's translation? Translation is the second part of the central dogma of the molecular biology. It's from RNA to protein. Okay, when the RNA just reaches the ribosome, then the mRNA comes, and this is the mRNA, this blue things. They bring one amino acid here and decode the code, like the UUU. They bring an AAA, and then like this amino acid become a protein, and then we can use the protein. But the code, we know there's 4 basic codes: A, C, G, T, or U. Okay, and in and we know that every 3 letters is called a codon, like that, like a CCU, CUG, UCA, UCC, like that. And 3 of them are a codon, but there's some stops. Like if it's AAU, they will stop. No, not AAU, UAA. Okay, if it's a UAG, it will stop, or if it's a UGA, it's a stop. And all the things share the same codon, and both of them will use this chart. That means that they come from the same, their ancestors are same and share the same evolution. And when this translation work, we know that they just bring the amino acids here, and then they bring the anticodon here, like this UUU. The anticodon will be AAA. Put together and then just form a protein.